Okay, hopefully we can share slideshow. Okay. All right. So, right, anatomy students, you are having to take a test on the cardiovascular system on Wednesday. Okay, so right before, I think it's right before lunch, which is good for us because, some of us go in, we run over. Um, anyway, so this is me like kind of recording for people who are absent, kind of the back third, fourth, who knows, of the lecture over that Thursday and Friday, so that y'all can I can kind of explain things. What did I talk about? What did I say? Anyway, here we go. And I'm going to kind of go faster because I don't have all of the students around, and pardon my hair because it's hot in here and I had to get it out of my face. Anyway, let's go. All right, so the heartbeat. Right before this, right, we ended with myocardial infarction. So right now we're going through the conducting system. So basically, right, the heartbeat is multiple contractions of individual cardiac muscle cells, okay, that have to occur in a specific sequence. We're going to have two major players to that. So two major types of cells. The first one being the contractile cells, which actually do the mechanics of contraction and propel the blood, okay? And then we have the conducting system cells, which kind of coordinates all of that movement. So in class, I came up with a ton of different analogies. And the one that I think kind of finally stuck is, so we have like a coach, and then we have our players. So the coach that kind of controls and coordinates things, and then the players who actually like play the game. In this case, for the heart, right, we have our conducting system cells that are going to coordinate that electrical movement. Then we have the contractile cells that actually are going to do the mechanics of the pumping. So contractile cells are the majority. They make up about 99% of all cardiac muscle cells. This makes sense, right? Because our heart is a pump, and that pump has to work for, some people live to be 115 years old. So a really long time, right? We've already talked about this. So the steps, there's going to be 3 steps. Like skeletal muscle, this tiny little corner up here. Let's see. All right. That's so bad, but that's okay. And then we have, okay, depolarization and repolarization and all of those things, skeletal versus cardiac. Okay. Now, step 1 in cardiac muscle is just like step 1 in skeletal muscle contraction. Okay. So we have rapid depolarization. Depolarization happens because of the influx of sodium, okay, into our membrane. And then those channels for cardiac, though, is going to close whenever that cell reaches a membrane potential. Remember, it's millivolts on our y-axis and in seconds on our x. Okay, around positive 30 millivolts, okay? Then the one that's the difference, like I told everybody in class, like star this one, box this one's different. It's called the plateau. And the plateau is super important, okay? So we've excited our sodium channels. We have an influx of sodium, so we have rapid depolarization. Now, during the plateau of cardiac cells, okay, we have, they're actually beginning to pump sodium out and replace it with calcium. So we're pumping sodium out. We open our voltage gated calcium channel. So voltage gated means it has to reach a certain voltage, and the voltage previously was this positive 30 millivolts. Okay, so once it reaches that positive 30 millivolts, we're pumping sodium out, and then we've opened those voltage gated calcium channels. So we're kind of exchanging sodium for calcium. Okay, and so that extracellular calcium enters the sarcoplasm. Sarcoplasm, cytoplasm, that gel-like stuff inside the cell, okay? Which is where like half of cellular respiration happens. If anybody remembers, thinks back to our biology and chemistry chapters. Okay. So this plateau and those channels kind of remain open for a relatively long time, okay? And that balance, the loss of sodium and that influx of calcium, allows a cardiac contractile cell to delay repolarization. So in the skeletal, right, we have rapid and then rapid. We have rapid depolarization, rapid repolarization. We don't want that in a cardiac muscle cell. We have a delay. Okay, so it kind of stays the same. We have no change in voltage. It stays around 0 millivolts. Okay, and this is where the initiation of the contraction happens. Right, so here, right, we have rapid. I'm pointing to like this little thing here. Okay, and then right, transient outward flow of potassium, that's extraneous. We're going to talk about potassium here just with repolarization. Okay, so we have opening of voltage gated calcium channels, and so we're remaining kind of neutral at 0 millivolts. Okay, so then we have the quintessential repolarization, which means we're closing calcium channels, we're opening potassium, potassium rushes out of the cell, and so we're back to our resting membrane potential. So really for cardiac, the only step that is different is step 2. Okay, so the action potential of a cardiac muscle cell is prolonged. Remember, it's like pauses and it takes a little bit more time, is prolonged because of calcium. And the period of contraction actually ends when repolarization happens. So contraction is occurring, and then relaxation starts when repolarization starts. This whole process of all the steps lasts around 250 to 300 milliseconds. All right, and then, right, I'm trying to have y'all compare skeletal versus cardiac because cardiac is going to be slower. Skeletal muscle depolarization and repolarization, hyperpolarization, happens around 25 to 30 times faster. Now, the reason that this is good for our body, this is good for homeostasis, is because this means that cardiac muscle tissue cannot tetany. Now, remember, I talked about in the skeletal muscle system, tetany, you could have complete or incomplete. And it's actually, don't confuse it with these graphs, okay? You have actual like contraction, force of contraction. And it can happen because of stimulation, like overstimulated. I don't know if you've ever like worked out and you have little pulses of contractions. Like I've certainly felt that whenever you kind of push your body over the limit because you have like residual electrolytes kind of trying to balance. And what happens with tetany is you over excitation and then it clenches and it does not relax. That's bad, okay? Because that can lead to muscle atrophy, okay? But because we have that plateau in cardiac muscle, there's no overstimulation. There's no overstimulation. And so that calcium influx allows it to last longer, so a contraction fully ends before repolarization happens, eliminating the potential for tetany. So cardiac muscle tissue cannot tetany. That refractory period, that plateau, is the reason why. It's very essential. It's very important. Calcium is going to be super essential. Yeah, so here, let me move myself out the way. All right, so, right, so we have don't get this confused, read your axes. So on the Y, we have force of contraction, okay, over time on the X. So, right, we have a little bit of force and then none at all, which means it's relaxing. Force of contraction and then a little bit and then another one because of that stimulation. And then we go all the way back to relaxation. And then a little contraction, a little contraction, a little contraction, a little, and then we go all the way back down. That is incomplete tetanus. And then we have complete up here, which is bad. Okay. So, right, whenever we're overstimulated, that can lead to complete tetany where it clenches, it does not relax. Okay. So also, let me just again move myself over here, down here, okay, is we can compare that rapid repolarization of skeletal muscle, repolarization on the back end, okay, to that only happens, number 3 is the repolarization after the plateau of cardiac. So, right, this one looks like skeletal muscle. The plateau is unique and essential to cardiac muscle tissue. So I might be able to recognize the graphs, right? We have different types of steps for skeletal muscle, but the main one that allows us to not tetany is the plateau. Okay, so then I talked about how that depolarization, plateau, repolarization kind of is going to tell us information whenever we talk about a regular pulse wave. So this is a pulse wave. Right here, this first initial hump is called the P wave. P wave, atrial depolarization and contraction, we call that systole. So atrial systole happens with the P wave, okay? Then we have actual ventricle contraction or ventricle systole in the QRS complex. So this like spiky looking thing is the QRS complex, okay? And if you notice, it happens during the plateau, which makes sense, right? I just talked about how a full contraction will happen, okay? A full contraction will happen until the very end of the plateau. So whenever we repolarize, number 3 in this case, we have ventricle diastole, which means relaxation. So I don't know if you can tell, but this T wave, this back T wave, which tells us ventricle diastole, is at the look, we can line it up. It's at the very end of that plateau, okay? So pulse wave, right, we have atrial systole, but in reference to all of the electrical signals, we care about the atrium. It's important. It's the filler, okay? But it's to make sure that the ventricles are loaded. So we really are look at how all of this, the QRS complex and the T wave, happen within that plateau. So essential ions, whenever we're relating things to nutrition, right? I said heart blockage is caused by bad cholesterol. We also need to make sure we're ingesting calcium because calcium is going to allow that plateau to happen so that we don't tetany. Now, another thing that I want to look at this graph is the classes of drugs. So I said that whenever we have heart rate, heart rhythm, so all of this pulse wave is a heartbeat, heart cycle, that is directly related to blood pressure. A high heart rate usually indicates that we have high blood pressure. And then I'm going to talk, right, I talked about systolic versus diastolic blood pressure. That top number is the one that I care about because it's talking about actual contraction, which means the pushing pressure. So all of these classes of drugs, I have a sodium channel blocker, I have a calcium channel blocker, I have a potassium channel blocker, and then I have something called a beta blocker. Okay. All of these are going to be medications that I can give to a patient that can help lower their blood pressure, raise their blood pressure if they have low blood pressure to begin with. Okay. Um, Also, not on this class, but you can use diuretics. So making somebody pee a lot is going to also help lower their blood pressure. But Yao had a good question in class. The difference between classes, so right, those were classes, versus schedules. So I inserted this slide. So here is the USDA drug scheduling. Now this is subject to change, I think, in every single state. So right, the FDA, we have cannabis technically could be called a supplement because it's a plant, but there's no really accepted medical use, which is why it's under a schedule. So if you look at these, right, schedules mean they have potentially high dangerous effects for patients who use them. So right, Vicodin, codeine, heroin, LSD, all of these, right, are probably drugs that you've heard of that um, are addictive and just cause like a lower quality of life whenever you're hooked on them. So we have them under different schedules. Now, the ones that are not right, so we can read this um, in the green, right? We have drugs with lower potential of abuse. Okay, so Robitussin. So, right, sometimes you have to show your ID to get like cough medicine in just a grocery store. So that's kind of like these drugs in Schedule 5. Now, Schedule 1 and Schedule 2 are the ones that are going to be high potential for abuse. And the only reason that we have Schedule 2 is that there are some uses. There are some. So Vicodin is a painkiller. There are some instances where Vicodin is the only pain medicine that we probably could use, okay? Instead of like morphine or fentanyl or tramadol or any other things, right? Vicodin. Cocaine is a vasospasmal drug, so it can cause nosebleeds, but it can also stop nosebleeds. So there are some instances where cocaine can be prescribed in a hospital setting, and meth. So there is like some reason for it. Anyway, those are schedules different than classes. So classes are just going to talk about the actual chemical mechanism of a drug. So we have class 1, they're highly soluble, high permeability. All right. Class 2, low versus high. All right. So that actually talks about how the mechanism of the chemical of the drug versus the schedule. Okay, so difference between classes and schedules. Anyway, let's get to the conducting system. So the conducting system, right? The heart contracts on its own in the absence of neural and hormonal stimulation. That is because we have pacemaker cells, which allow for the heart to have this thing called automaticity. It can run by itself. Okay. So we have those specialized cells that can initiate and distribute those electrical impulses, and those are called nodal cells. Nodal cells. So we have one main pacemaker, we have a backup pacemaker, and then technically I told them, because in the video there is a third pacemaker cell, third like wave of pacemaker cells, but if you're on your third pacemaker, that it's not going to work. It's just not going to work. Okay. So we have our nodal cells. So we have the first one, all right, which, right, these are all responsible for enabling the rate of contraction, setting the pace of the rhythm. We have sinoatrial, or the SA node, SA nodal cells. Okay. These are cells that are conducting the action potentials and the electrical impulses. Then we have the atrial ventricular. So the sinoatrial is going to be just in that atrium versus the ventricle. This makes sense because what has to contract first? The atria. And then the AV, atrioventricular, just like in the name, it's going to sit in between the atrium and the ventricles. So these nodal cells are going to depolarize spontaneously. They're going to generate an action potentials at regular intervals, which are going to determine our heart rate. Okay. And we're going to talk about like which ones have resting heart rate, which ones are a little bit lower in the next slide. Okay. So this is, I'm moving myself all around. Okay. So this is an image. So right here in the atrium, we have these fibers of cells, okay? And the SA node sits right here. We have the AV node. It goes down the interventricular septum. The actual like bundle of fibers, okay, is going to diverge. We call that the bundle of Hiss, Hiss. All right, and then it goes down into these branches called Purkinje fibers, okay? And I talked about how this is super important because those fibers are actually going to contract from the bottom up, all right? So in this conducting system, right, I talked about pacemaker cells. So the nodal cells, right, are going to reach threshold first. These are pacemaker cells. So at the SA node, this means that that electrical impulse is going to allow us to reach around 70 to 80 action potentials per minute. Now, those action potentials per minute are going to be a direct correlation to beats per minute. So 70 to 80 beats per minute. Action potentials per minute equals beats per minute. So at the SA node, we have set 70 to 80 beats per minute. Now, 70 is kind of closer to resting, okay? Whenever we're getting up and doing kind of light activity, we can reach about 80 to 90, okay? So SA kind of covers resting to activity. All right, so the stimulus has to be distributed so that the atria contract together and before the ventricles. So all of that atrial contraction has to happen before the ventricles. And then the ventricles have to contract together. So the contraction, this is important, I would highlight this. The contraction happens, oh, that doesn't even make sense. Contraction begins at apex. Okay. The contraction begins at the apex. So we have the apex right here of the heart, okay? And how these are oriented is why that contraction is going to start at the bottom. And I talked about why this is important because if we start at the top, right, we're at risk of not getting all of the blood out. If it happens at the bottom, we have a complete squeeze out to our semilunar So the cells of the SA node are electrically connected to those of the AV node. Right? So it's all about carrying the signal. So the signal happens in the atrium first. The atrium are going to contract. Then it travels to the sinoatrial node, which means like, hey, the atrium have already received the signal. You should now contract. Okay. So those conducting cells are embedded in the atrial wall, which I don't know if you can really see on this picture, okay? And the AV node is our backup pacemaker. So our number one pacemaker cell is the SA node, okay? Number one pacemaker, sets the pace. Then we have our backup at the AV node. And the AV node, if that one is the one that receives the signal first, it's going to make around a rate of 40 to 60 action potentials per minute. And I said that that's directly beats per minute as well. So 40 to 60 beats per minute. Okay. And that one is at the lower end of rest. It's still able to make things contract, but it's going to be a lot slower, all right, because our primary pacemaker cell is not working, okay? Both are going to work together, okay? But the SA node, if it's fully functional, is going to set the pace first. The AV node is just going to receive that same signal, okay, and do the same thing that the SA node is doing. If the SA node is not working, it's not able to conduct that electrical signal, the AV node will kick in and set the pace. Okay. So I just wanted you to see like the purple shading of what happens. So we have the sinoatrial node, and then the atrium contract, and then it travels down. The action potential travels down the bundle of His to the Purkinje fibers, and then from the bottom up, from the apex up in the ventricles, we contract, right? So that we can expel all blood. We don't have any blood that's sitting and pooling and at risk for clotting. Okay. So now let's go to the conducting system again. All right. Ooh, I have our EKGs over here. So the conducting system continued. At the AV node, the impulse is going to slow down. The impulse is going to slow down around 100 milliseconds to pass. This allows all of the things that the atrium need to do to happen. So that delay is important, right? Because blood has to be moving, the atrium have to contract, right? All of that has to happen first before the ventricles even start contracting. We don't want them to contract at the same time. That is why at the AV node, atrioventricular, it's going to slow down. So the atrium have all of that time to do a complete contraction cycle. Okay. Yeah. So we can have with the nodes, those electrical impulses at the nodes that are directly related to heart rate. Okay. So we have action potentials per minute equal beats per minute. If we have a slow one, the 40 is low. 40 is super low. All right. I would want you to raise that. That is called bradycardia. Okay. So a slower heart rate than 60 beats per minute, because 60 is resting. Okay. You have bradycardia. Now, a lot of y'all have asked me about like athletes. Some of them probably could live a little bit lower than this. I think like resting could be around 56. But yeah, 40 is going to be too low, but it's all about baseline, okay? Because some people can have more of a tachy heart rhythm, and this is going to be higher than 100 beats per minute. Now, whenever you're exercising, you have sinal tachycardia. It means you're still able to fully contract. It's just your heart is trying to keep up with the demand of oxygen whenever you're working out. Okay, so tachy is faster than 100 beats per minute. Boop. Okay, so we can actually have pictures of this in what we call an electrocardiogram. Now, this is a 5-lead electrocardiogram. One that you might see in the hospital is going to be 12-lead, but actually, y'all don't really need to know that, just in case you wanted to know. But this is 5 leads. So an ECG or EKG electrocardiogram is going to be an actual picture of the electrical currents happening in the heart. It's so powerful that we're able to detect them via electrodes on the surface of the body. Now, we see this picture in terms of systole versus diastole, which is contraction versus relaxation, systolic versus diastolic, contracting pressure versus relaxing pressure. On an EKG, it's contracting electrical current versus relaxing electrical current. So whenever we read those graphs, like on the previous slide, we're able to detect arrhythmias. So the first one is going to be the P wave, And that P wave mimics depolarization of the atria. And about halfway through, okay, contraction of the atria occurs, all within the P wave. So that little first blip, P wave, depolarization of the atria, atrial systole. And then we have the QRS complex, which means that the ventricles are going to depolarize. The ventricles contract shortly after that peak of the Q, right? We have QR, okay? So depolarization, all of that. So look, we have atrial diastole, atrial relaxation until after we've depolarized. Whenever we've depolarized because of the plateau, we have contraction, atrial systole. Then we have atrial systole until, that R of the QRS complex. And then, look, atrial diastole. All this time is atrial diastole because we're going to go down to this bottom section. We have ventricular systole, which is contraction, right at that P, or right at that R. And then we go to T, the T wave, which is ventricular diastole. So let's continue, right? T wave, ventricular repolarization. And I said that the ventricle is going to have a stronger electrical current, so we can't even pick up atrial repolarization because the ventricles are going to have like a stronger signal in this case. And there are two arrhythmias that I wanted y'all to know. AFib, which is the chaotic like heart signaling, all right, uncoordinated heart rate. You can live with this one. It's serious. You might go in for treatment, but you can live with it. You can live in AFib. The one that is deadly is VFib, which means like it's the fluttering that my sister talked about, that quivering, okay? So that electrical signal causes them to not fully contract, it just flutters, and this is bad. It's the most dangerous and it leads to death, right? In the atrium, right, since it's loading the ventricles, I just dropped something. It's loading the ventricles, it's going to be less serious than whenever we're actually propelling blood to all of the places that need the oxygen, including the heart itself, right? So here is a depiction of atrial fibrillation, and I said, can you kind of pinpoint a P wave? Sorry, I dropped something. Where it went. Okay. And the answer is no. It's not really distinguishable. I can see kind of little ones, maybe, but they're not super clear and they're not very pretty, right? And that's AFib because it's not fully contracting. The P wave in a heart rhythm tells me atrial depolarization and atrial contraction. And this shows me that I'm not having a good contraction, especially right here, right? I have little to none. Okay, and then I have an atrial flutter. Atrial flutter means that my contraction isn't really fully happening and it's fluttering. It's trying. There's electrical signal in the atrium, but it's not enough to actually fully contract. And then, right, I have a normal one. We can see the P, QRS, T, P, QRS, T, normal sinus rhythm. Then I have VFib, and it looks bad. You can't distinguish anything. You can't distinguish a P wave, QRS, T. It's just bad. Chaotic rhythm, it's fluttering. No blood is actually being propelled to maintain blood pressure and to get oxygen to the body, which are the two main functions, remember? So I had a video. I'm going to skip through these. I'm skipping through the compressions too because we're going to do that in class.
Our heart is an amazing organ.
And I'm going to go through heart dynamics and I'm almost finished. I think I can do it in 5 minutes. Okay, so heart dynamics. Okay. Heart dynamics is going to refer to the movement and the forces generated during cardiac contractions. I'm going to care about two. So the forces and the movement. I only care about two. The first one is going to be stroke volume. Stroke volume just means the amount of blood ejected by a ventricle. All right? The amount of blood ejected by a ventricle. This can vary beat to beat. Okay? The main one that I'm going to care about whenever I'm thinking about heart dynamics and looking at somebody, are they able to maintain homeostasis, is going to be cardiac output, or CO. Cardiac output is the amount of blood pumped by each ventricle in a minute. Now, I said that there's going to be a little bit of math involved in this, okay? So cardiac output is pumped by each one in a minute, okay? It's going to use the stroke volume in order to calculate it, Okay, and the reason that cardiac output is more important is because it indicates that blood flow through peripheral tissues. Okay, so the formula for that is going to be cardiac output is equal to the stroke volume times the heart rate. And I said it's going to be very simple. So let's say that my stroke volume, the amount ejected by each ventricle, I'm going to have to use the average, right? Because it can vary from beat to beat. I want to use an average, is around 80 milliliters per beat. Okay, and my heart rate is 70 beats per minute. What is my cardiac output in liters? Okay, so remember 80 times 70 is around, it's not around, it's exactly 5,600 or 5,600. Okay, but that's milliliters. That's milliliters. I want it in liters. So milliliters to liters, there's 1,000 milliliters in 1 liter. That means that my cardiac output is around 5.6 liters. Okay, in a minute. That is normal. A normal average adult human is going to have a cardiac output of around 5.6 liters. If it's lower than that, that indicates that something's wrong. So that's why cardiac output is going to be the major one. Okay, and we're almost here. This is the last 2 slides. Okay, so I have human lung anatomy. You don't need to do any labeling. I just wanted to arrow in on the fact of that gas exchange. So we talked about going to the pulmonary circuit. Lungs are the pulmonary system. We're going to breathe in, and that oxygen is going to travel down our trachea, down to our bronchus, down to our bronchioles, down to our alveoli. And our alveoli are going to be surrounded by a blood vessel. You guessed it, the capillaries, okay, that aid in that gas exchange. So here's another depiction. So it goes down through our lungs. And the only thing I wanted y'all to know in that circuit is whenever we have the arrow to the lungs, now we're going to add another arrow to the capillaries surrounding the alveoli. Okay, the capillaries surrounding the alveoli, because right, those capillaries, those blood vessels are going to be able to exchange carbon dioxide for oxygen and vice versa. It's going to be able to take carbon dioxide and give oxygen, and transport it back through veins to the heart. So the capillaries are our target blood vessel in the pulmonary system. And that's pretty much the end of the cardiovascular system. So hopefully me kind of talking through it helped. Let me know if you'll have questions, and hopefully this recording worked. Let me stop sharing and let me end that recording. So fingers crossed.